Question 3. It shows you figure 9. Okay, so the carbohydrate in figure 9 is formed from two hexoses sugar. So hexoses are 6 carbon sugars. Okay, so examples of 6 carbon sugar, you have alpha glucose, beta glucose, you have the galactose, and also you have the fructose. Okay, so those are the monomers that has 6 carbon sugars with 6 carbon. So you have to know, okay, how to number the carbon in order for you to be able to know the type of glycosidic linkage. So in this case, the structure shown here is a disaccharide, which is sucrose. Okay, so how do you know that it is sucrose? Because the monomer for sucrose consists of alpha glucose and also fructose. Okay, so therefore in the diagram here, the first monomer here is alpha glucose. How do you know it is alpha glucose? Because at carbon number 1 here, the hydroxyl group is located below the plane of carbon number 1. Okay, and then for fructose, the structure seems that the sugar consists of 5 carbon sugar, but it is actually 6 carbon sugar. Okay, so for fructose here, the carbon number 1 start here. Okay, carbon number 1. Okay, so carbon number 1, carbon number 2, number 3, number 4, number 5, and number 6. As for the glucose, carbon number 1, number 2, number 3, number 4, number 5, and number 6. Okay, so you really have to know how to number the carbon for the hexose sugar, okay, or any type of sugar. So identify this carbohydrate. So the answer will be disaccharides. Okay, so disaccharide, or to be specific, it is sucrose. Okay, you can answer either one. The disaccharide, okay, or sucrose. Okay, because specifically shown here is the structure of a sucrose. Okay, so name one function of a carbohydrate shown. Okay, so sucrose or disaccharide, any disaccharide can be other types of disaccharide. You can have maltose and also lactose. Okay, maltose, lactose, and sucrose. So those are the type of disaccharides. And basically, the function of disaccharide is that it function as energy storage. Okay, energy storage. Okay, because when the disaccharide is broken down into its monomer or undergo the process of hydrolysis, okay, so it breaks the molecule back to its monomer. So the monomer consists of glucose and fructose, for example, in this case. So the monomer can be used during cellular respiration to generate ATP in the form of to generate energy in the form of ATP. So that's why the function of disaccharide is that it functions as energy storage. Okay, storage of energy. Figure 10, question B, is a fibrous protein. Okay, so as you can see here in this diagram, it is the structure of collagen. Okay, so collagen. Okay, so this is a structure of a collagen. Okay, so collagen is basically a type of fibrous protein. Okay, so as you can see from the structure of the collagen, it consists of 1, okay, 2, 3. Okay, so 3, basically it consists of 3 identical helical polypeptide. Okay, polypeptides that is coiled into a larger triple helix. Okay, coiled into a larger triple helix. Okay, so this is the structure of a collagen. And then basically the structure of collagen allows the collagen to be really strong. Okay, it has a great strength. So the function of collagen is basically it forms it function as girders of connective tissue in skin, bone, tendon, and also ligaments. Okay, so collagen will account for 40% of the protein in the human body. So in the question number 1 for B1, draw the basic monomer of X and name one of its functional group. Okay. Okay, so as you know, the monomer for protein is amino acid. Okay, so you have to draw the correct structure of amino acid, like we have seen in question 2. Okay, so you have to know how to draw the correct structure of amino acid, where the carbon, you have the carbon that is bonded to hydrogen, and then you have the nitrogen. Okay, the amino group. And then the carbon is bonded to a side chain, and then you have the carbon that is bonded to the carboxylic acid. Okay, so this is the monomer for the basic monomer for protein, and name one of its functional groups. So as you can see in the structure of this amino acid, it has 2 functional groups. So the first one here is the carboxyl group. So you can label, name this as carboxylic acid. Okay, carboxyl acid, carboxyl group, sorry, it is carboxyl group. The compound is carboxylic acid. Okay, and then this one is amino group. Okay, so this is the basic structure of an amino acid. If, for example, the question asks you to draw the Zwitter ion form, so you just remove the hydrogen from the carboxyl and put a negative sign at the oxygen. And then, for example, here, again, if the question asks you to draw the Zwitter ion form, okay, so you just for the amino group, you add another hydrogen ion, so you put a plus sign at the nitrogen, so that is in the Zwitter ionic form. Okay, and then the hydrogen, and then the side chain, and then the carboxyl group will be ionized to become COO negative. Okay, so make sure you put the charge correctly at its places. So the plus sign is at the nitrogen, the negative sign is at the oxygen. So this is the Zwitter ionic form. So the correct answer will be this one lah, because the question just wants you to draw the basic structure of amino acid. Okay, number 2. Describe the level of organization of the protein shown in figure 10. So basically, as you can see, the collagen consists of 3 polypeptide chain that is coiled together. So it consists of more than 2 polypeptide that are joined together. So when more than 2 polypeptide join together to form a functional protein, so obviously it is in the quaternary level. Okay, quaternary level. Quaternary structure. It is a quaternary structure. The question with 2 marks. Okay. Okay, so you have to describe. What is quaternary structure, where quaternary structure, as I mentioned, it consists of 2 or more polypeptide chains, okay, joined to form a functional protein. Okay, 2 consists of 2 or more polypeptide chains. Okay, number 3, name one example of fibrous protein. Okay, so you have one type, which is this one, collagen. Okay, so other types of fibrous protein, there are keratin. Okay, keratin. So keratins are proteins that are found in your hair, for example, in your skin. Okay, and then other types of fibrous protein, you can have elastin. Okay, elastin or fibrin. Okay, which is protein found in silk. Okay, C. Name the process of converting unsaturated fats to saturated fats. So unsaturated fats is basically fats in liquid form. Okay, fats in liquid form, for example, vegetable oil. Okay, vegetable oil, and also fish oil. Okay, so those are fats that is unsaturated, where at room temperature it is in liquid form. And as for saturated fats, there are animal fats at room temperature, they are in solid form. So name the process of converting liquid. For example, you have vegetable oil, you want to make a margarine, for example. So the process of converting unsaturated is to saturated fat is hydrogenation. Okay, so as you know, in the structure of triglyceride, the fatty acid molecule, it has double bond, okay, between the carbon. Okay, double bond between the carbon, so it causes the molecule, the triglyceride molecule, to be bent. Okay, bent, or it has kink. Okay, where the double bond exists, so it causes the fatty acid molecule to be bent. So due to the bent structure of triglyceride, so this prevents the triglyceride molecule from packing close together, and this prevents the molecule to form solid. So therefore this type of fat, they are liquid at room temperature. So the bent structure of the fat causes the fat molecule to not be able to pack closely together. And as for saturated fats, okay, because all the carbon is has hydrogen atom bond to the carbons, so it causes the fat molecule to be straight. Okay, straight molecule. The triglyceride are straight molecules, so that's why at room temperature, okay, so they exist as solid because the molecule of fat can pack closely together to form solid. Okay, so okay, so that is for C, the explanation for fats. Okay, so the answer will be hydrogenation. Okay, B. Name the components that make up nucleotides. Alright, so nucleotide, as you know, it is the monomer for polynucleotides, DNA or RNA. So the structure consists of phosphate, okay, that is bonded to a 5-carbon sugar. Okay, the 5-carbon sugar or a pentose sugar. Pentose sugar, okay, and this sugar is bonded to a nitrogenous base. Okay, a nitrogenous base. So basically the structure of a nucleotide, if I can draw it here, so phosphate bonded to a 5-carbon sugar, and the sugar is bonded to a base. Okay, so the base can be adenine, guanine, thymine, cytosine, and uracil. Okay, so this is an oxygen atom. Okay, and then the carbon are numbered as 1, 2, 3, 4, and 5. Okay, so that is the structure of a nucleotide. E. How many hydrogen bonds are found between thymine and adenine? Okay, so along the chain of DNA molecule, along the structure of DNA molecule, so where the sequence has adenine, so adenine has to pair with thymine. Okay, so in order to hold the in order to hold the DNA strand together, hydrogen bonds has to form between the bases. Okay, hydrogen bond has to form between the bases. So if the base involve adenine and thymine, so the bond form is 2. Okay, there will be 2 hydrogen bonds formed between adenine and guanine. And as for the next pair, guanine and cytosine, for example, okay, guanine has to pair with cytosine. So there the amount of hydrogen bond formed between guanine and cytosine, okay, there are 3 hydrogen bonds formed between guanine and cytosine along the DNA strand, okay, along the 2 DNA strand, along the 2 complementary DNA strand. Okay, so the answer for E will be 2. Okay, 2 hydrogen bonds formed between adenine and thymine. Okay, question number 4. Figure 11 shows water molecule. Okay, so as you can see here, one water molecule, okay, consists of hydrogen atom and 2 oxygen atom and 2 hydrogen atoms. Okay, so where the region has the oxygen atom, there will be a partial negative charge. Where the region has a hydrogen atom there, okay, so there will be partial positive charge. Okay, so in this question, identify A. So A will be hydrogen bond. Okay, hydrogen bond. Okay, so the interaction between water molecules will be hydrogen bond. So as you can see, the hydrogen bond is formed between hydrogen atom of one water molecule to oxygen atom of another water molecule. Okay, so the attraction is between partial negative charge of oxygen and partial positive charge of hydrogen atom, okay, of different water molecules. So this produce a weak interaction between the water molecule, which is hydrogen bond. Okay, number 2, explain the property of water when it changes from liquid to ice. Okay, so explain the property of water when it changes from liquid to ice. Okay, so when water changes from liquid to ice, basically for a water molecule, one water molecule, okay, will formed 4 hydrogen bonds, okay, to 4 other water molecules. Okay, so that is the first point. Okay, second point, okay, because the formation of hydrogen bonds between these water molecules are fixed, okay, fixed, the hydrogen bond will not break as it will be in liquid form. In liquid form, water molecule, the hydrogen bonds between water molecule will keep on breaking and reforming. That is in liquid state. Okay, you have a high frequency of hydrogen bond to break and reform. But when the water is turning to ice, okay, in solid form, the hydrogen bond between the water molecule will be fixed. Okay, one water molecule will form 4 hydrogen bond. Okay, one water molecule form 4 hydrogen bond with another water molecule, for example here. Okay, so this is to form a crystal structure, okay, a strong structure, or this form a more stable hydrogen bond. So that is for the second point. Okay, to form a rigid or a hexagonal or a crystal structure. Okay, if I can write this, to form a rigid, okay, a rigid or hexagonal or crystal structure in the ice, okay, or to form a strong structure, or to form a more stable a more stable hydrogen bond. Okay. So that is the 2 points that you can get. Okay, the 2 marks. Okay, the explanation. Or you can add another point there, is that when water changes from liquid to ice, the ice will expand. Okay, expand when it freezes. Okay, the ice will expand when it freezes. Okay, expand when it freezes. Or ice is less dense. Okay, ice is less dense, or it has a low density compared to water in liquid form. Okay, so that's why ice can float in water. Okay, so next, B, figure 12 shows a dipeptide. Okay, so here you have a dipeptide bond. Sorry, you have a peptide bond. The molecule is dipeptide. Okay, so the bond is peptide bond. Label the peptide bond is there lah. I've already drawn there. Okay, so label the peptide bond will be there. So 1 mark, so easy. Number 2, draw 1 amino acid of the dipeptide. Okay, so you have to know that it consists of 2 amino acid. So the first amino acid will be here, and the second amino acid will be here. Okay, so you just draw 1 of the amino acid correctly. So if you decide to draw the first amino acid, so at the carbon here, you just add another OH group. Okay, so for example, nitrogen, carbon. So you draw exactly what is given in the question. Okay, so alright, so the carbon is bonded to a hydroxyl. So this is the first amino acid. If the dipeptide were to be broken, okay, into its monomer through the process of hydrolysis. Okay, hydrolysis. Or you can draw the second amino acid. Okay, the second amino acid will be okay, where you have the nitrogen, you add another hydrogen. Okay, and then just complete the drawing like that. Okay, so you just draw exactly what is given in the question. Okay, so that is 1 mark. So you have to draw it properly. So for example, if you were to change the side chain here, okay, you were to add you just write R there. Okay, so your answer would be wrong. Okay, so you draw exactly what is given in the question. You just break it down into the separate amino acid. Okay, so number 3, identify the group of amino acid that B belongs to. Okay, so as you can see, the amino acid, its side chain consists of methyl group. Okay, so methyl group consists of carbon and hydrogen, so it is hydrocarbon. So hydrocarbon is the property is nonpolar. Okay, so therefore the whole amino acid here will be a nonpolar amino acid. Okay, a nonpolar amino acid or hydrophobic amino acid. So that is the answer for 3 here. And then number 4, protein can be classified according to their function. Give 2 examples of protein with their respective function. Okay, so you have so many different types of protein, example of protein and their function. So if you look at your notes, okay, slide 83, in the table in slide 83, it gives you several different functions of protein and the description for the function of the protein. For example, protein can be enzyme. Okay, it can be enzyme, and the function of the enzyme is to involve in catalyzing chemical reaction. So that is 1 mark. Okay, another function of protein, it can function as defense protein. Okay, such as antibody or immunoglobulin, and the function is to protect your body from infection of pathogen that can cause disease. Okay, and then another example of protein, okay, another function of protein is that it can function as storage protein, such as albumin, okay, to store amino acid, okay, albumin that is found in eggs, okay. And then another function of protein, it can be as transport protein, okay, transport protein that are found embedded in the plasma membrane, for example, which controls the movement of substance in and out of the cell, okay, transport protein. And then you can have protein that function as hormones, okay. So there are proteins that are made up, sorry, there are hormones that are made up of protein, such as insulin, okay, that is the function of this hormone is to coordinate the organism responses, okay, to coordinate the function of that organism, okay, the body function, to coordinate a mechanism, okay, within an organism. And then the next type of protein is the protein can function as receptor proteins, okay. So this receptor protein, for example, it can be found embedded in the plasma membrane of protein, which function to receive signals, okay, receive signals from molecules outside of the cell, okay. Another type of protein, you can have motor proteins, okay, which function in cell movement, okay, contraction of muscle, such as the interaction between actin and myosin, okay, motor proteins, for example. And then the last one, you can have structural proteins, such as the one that we looked at previously, such as collagen, keratin, okay, structural protein that give structural support, okay. So those are the different type of protein that you can write. So you have so many, so you just mention any two. Okay, number 3, question number C, okay, question C. Figure 13 shows the structure of a disaccharide, okay. So as you know, as you can see in this diagram, carbon number 1, okay, the hydroxy group is located below the plane, okay. Same goes to here, okay, carbon number 1 is below the plane. So identify why, okay, why you have to name basically the bond that is formed between the monosaccharides in the structure of disaccharide here, okay. So basically you have to know how to number the carbon lah, again. So this is carbon number 1, then only you can identify the type of glycosidic bond. So the first monomer here, okay, is alpha glucose. The second is also alpha glucose, okay. So for the second alpha glucose, the carbon starts here, okay, carbon number 1, number 2, number 3, number 4, number 5, number 6. The first glucose molecule, carbon number 1, number 2, number 3, number 4, number 5, number 6. Okay, so because the bond formed between carbon number 1 and carbon number 4, so the bond formed between the disaccharide, which is Y, is 1,4 glycosidic bond. Okay, or to be specific, you can also write alpha 1,4 glycosidic bond. Okay, so number 2, it says here amylose and amylopectin, okay, are the two components of starch. Describe the structure of amylopectin. Okay, so the basic structure, the basic knowledge you have to know is that amylose, the structure is helical. Okay, amylopectin is also helical, but it has branch. Okay, it has branch. Okay, so in the structure of amylose, it consists of monosaccharides. Okay, monosaccharide alpha glucose. The monomers, okay, for starch, let it be either, for example, amylose or amylopectin, the monomers are all consist of alpha glucose. So the alpha glucose are joined together by the 1,4 glycosidic bond, but to hold the helical structure is through hydrogen bonds. Okay, hydrogen bonds. And where you have the branches in the amylopectin structure, okay, this is amylopectin here, where you have branches, the bond will be 1,6 glycosidic linkage. Okay, so the question asks you to describe the structure of amylopectin. So basically, okay, so the first mark that you can get is that for amylopectin, it consists of alpha glucose monomers. One mark. Okay, describe the structure. It consists of alpha glucose monomer. Second, the structure of amylopectin is branched. Okay, it is made up of branches. Okay, branched polymer. Okay, it is a branch polymer. Okay, polymer. And then another point that you could add is that each branch is a short chain, okay, which is linked together by alpha 1,6 glycosidic bond. Okay, so the branches are held together by 1,6. So you can mention that point. Okay, the branches are held together by 1,6 glycosidic linkage. So those are the points that you could describe for amylopectin. Okay, question D. Figure 14 shows a structure of cellulose. Okay, so cellulose, the monomer consists of beta glucose. Okay, where at carbon number 1, the hydroxyl group is located above the plane. Okay, so as you can see, this is the first beta glucose, the second beta glucose, and the third beta glucose. If you look at the glucose number 2 here, okay, this glucose, the arrangement of the beta glucose is that they are arranged upside down, okay, to its respective neighboring monomers. Okay, so the first glucose will be organized upright. The arrangement is upright. The second monomer will be upside down. Okay, it will face downward. Okay, and then the third is upright, and so on. So they are arranged upside down with respective to its neighbor. Okay, so but the bond will still be, okay, between carbon number 1 and carbon number 4. Okay, carbon number 1 and carbon number 4. So for carbon number 2, the numbering will start from here. Carbon number 1, number 2, number 3, number 4, number 5, number 6. Okay, for the first monomer, carbon number 1, number 2, number 3, number 4, number 5, number 6. Okay, so the bond between the beta glucose in cellulose will still be 1,4 glycosidic bond, but because it involves beta glucose, okay, to be more specific, it is beta 1,4 glycosidic bond. Okay, so number 1. Okay, the question here is it asks you draw one monomer of the above diagram. So basically the question asks you to draw the structure of beta glucose. Okay, of beta glucose. So the answer will be just draw the structure of beta glucose lah. Okay, it is a 5 carbon sugar. Okay, so at carbon number 1, the hydroxyl group is located above the plane, so you have the hydrogen atom. So at carbon number 2, you have the hydroxyl group. Okay, hydroxyl. So make sure you place the hydroxyl group and the hydrogen atom correctly. Okay, this one is hydrogen, sorry, hydrogen, hydroxyl, hydrogen, and then this one you have the carbon number 6. Okay, CH2OH. Okay, so you have to know how to draw the monomers. Okay, the alpha glucose, the beta glucose, the structure of fructose. Okay, so you have to know how to draw them. Okay, so that is your answer. Okay, so make sure you place all the atoms correctly. Okay, so E. Nucleic acids are polymer of nucleotides that consist of 3 components. Okay, polymers, okay, three components of nucleotide. We know that it consists of phosphate bonded to a pentose sugar bonded to a base. Okay, so number 1, explain how DNA nucleotides are linked together by covalent bond into a single strand. Okay, so the bond between the nucleotides involved is a type of covalent bond, which is, to be specific, it is phosphodiester bond. Okay, phosphodiester bond. Okay, so in order to link together, okay, the nucleotides, it is through the reaction of condensation. Okay, so you have to explain how. Okay, so in order to link the nucleotides together is through the condensation reaction. That is for the first point. Okay, through condensation. You have to write the full sentence. I'm just giving you the point here. Okay, the keywords. Condensation reaction. So you have to spell it correctly. Okay, through condensation reaction. And then next, okay, a phosphate group that is attached to the carbon number 5 of a sugar, okay, okay, will join the phosphate group which is attached to carbon number 5 of the pentose sugar, okay, will form the covalent bond, or will join, okay, to the hydroxyl group which is located or attached to carbon number 3 of the pentose sugar of the next nucleotide. Okay, this is to form a phosphodiester bond between the two nucleotides, and the process involved the removal of a water molecule. Okay, so those are the answers that you could write down. So for example, if I were to draw it, okay, phosphate group. Okay, so you know phosphate is linked to a 5 carbon sugar, okay, and base. Okay, so at carbon number 3 here, okay, carbon number 1, number 2, number 3, number 4, number 5, here you have oxygen. Okay, so in order to join to the next nucleotide, okay, okay, you know that phosphate is attached to carbon number 5. Okay, so at carbon number 3 of the previous nucleotide, okay, at carbon number 3 it has hydroxyl. Okay, so in the reaction of condensation, this hydroxyl group at carbon number 5 has to be removed. Okay, it has to be removed. So you remove this, and then at the phosphate group here, there's OH. Okay, there's OH. Okay, so OH. So the hydrogen atom here and the hydroxyl group of carbon number 3 will be removed, okay, to form a water molecule. So in the process of condensation, a water molecule will be removed. Okay, removed. So that is one of the points that you can include in your answer. So so after you have removed a water molecule, a phosphodiester bond will be formed. Okay, so you just erase out, okay, and then you add a new bond there. Okay, so the phosphate group of the new nucleotide will join or form bond to carbon number 3, okay, of the previous nucleotide. So this is the bond, we call it as phosphodiester bond. Okay, this is a phosphodiester bond. Okay, so it involves the formation of phosphodiesterbond. Okay, so those are the points that that you can include in your answer. It wants 3 marks. It is worth 3 marks. Okay, so 3 points, 3 explanation there that you have to include in your answer. Okay, so number 2, name the nucleic acid that contains the nitrogenous base uracil. Okay, so the answer is obviously RNA. okay, RNA or ribonucleic acid. Okay, you can just write as RNA, or you can write the full name, which is ribonucleic acid. Number 3, ribose and deoxyribose are the pentose sugar of nucleic acid. Okay, differentiate between the two pentoses. So ribose is the 5-carbon sugar that is found in RNA. Okay, so the 5-carbon sugar here is ribose for RNA. But if for DNA, the 5-carbon sugar will be deoxyribose. So if you refer to your lecture note, okay, slide 136, it gives you the structure to compare between the structure of deoxyribose and ribose. Okay, so if you look at the structure of ribose at carbon number 2 of the sugar, it has a hydroxyl group, whereas for deoxyribose at carbon number 2, it has hydrogen atom. Okay, so that is the difference, the main difference of deoxyribose and ribose. You have to focus at carbon number 2. Okay, and so the question asks differentiate between the two pentoses. So basically ribose, okay, they are found in RNA. Okay, and as for deoxyribose, they are found or present in DNA. Okay, and the second, the question worth 2 marks, so you have to give 2 differences. So you have to describe for carbon number 2. So for carbon number 2, there is presence of hydroxyl group, and as for deoxyribose, at carbon number 2, there is presence of hydrogen atom. Okay, so question F. Figure 15 shows the formation of a triglyceride. Complete the chemical reaction, okay, by drawing the molecular structure in the box provided. Okay, so here we have looked at already in the previous question. Is basically glycerol. Okay, so glycerol, obviously in the box will be fatty acid to produce triglyceride, a molecule of triglyceride. So a molecule of triglyceride will consist of 1 glycerol and 3 fatty acid. So in this case, okay, the molecule that will be formed is, okay, here you have to separate the fatty acid molecule. Okay, you have to separate the fatty acid molecule. The carbon, okay, the carbon you have to add with a hydroxyl group. Okay, because now the triglyceride is broken down through the process of hydrolysis to produce back its component, which is 3 molecules of fatty acid and 1 molecule of glycerol. The reverse reaction will be hydrolysis. So you just draw the molecule for the 3 fatty acid, where the carbon here are, okay, R1 OH here, because this fatty acid, it has the carboxyl group. Okay, so that is the first amino acid, and then you just complete your drawing by drawing the other 2 fatty acid. Okay, so you just complete your drawing there to draw the 3 structure of the 3 fatty acid. Okay, so simple question. Right, so next question, why is butter solid while oil is liquid at room temperature? Okay, so butter is fat from animal. Okay, it's fat from animal. So at room temperature it will be solid because due to the presence of saturated fat. Okay, saturated fat, the component of saturated fat consists of saturated fatty acid, where the molecule of this fatty acid will be straight. Okay, straight molecule. So this causes the triglyceride molecule to be a straight molecule. Okay, so at room temperature, so they can pack closely together to form solid. Okay, whereas for fat that is in liquid form, okay, which is unsaturated fat. Okay, unsaturated fat consists of unsaturated fatty acid, where in the structure of unsaturated fatty acid it has double bond between the carbon, and where you have the double bond between the carbon, okay, so it causes the fatty acid molecule to be bent. So therefore the molecule of that unsaturated fat will be bent, okay, and therefore at room temperature, okay, because due to the presence of bend or kink, we call it as kink, where double bond occurs, this prevent the fat molecule from packed closely together. Okay, so they will be liquid at room temperature. It prevents the molecule of fat from closely packed together, and they will maintain as liquid at room temperature. Okay, so that is why butter is solid and unsaturated fat or oil or vegetable oil or fish oil is liquid at room temperature. Okay, so those are the points that you could write down. So there's many points that I've mentioned. I'm just going to repeat again the highlight of the answer. Okay, so first, butter is saturated fat. Okay, so the structure is saturated with hydrogen, or there is a maximum number of hydrogen atoms in the molecule of saturated fat. Okay, so there is no presence of double bond between the carbons in the fatty acid. Okay, and there is no kink. There is no presence of kink. So the molecule of saturated fat will be straight molecule. And as for oil, which is liquid at room temperature, it is unsaturated fat. Okay, the structure of the fat molecule or the fatty acid, which is the component of the unsaturated fat, has one or more double bond. Okay, has one or more double bond between the carbon, can accept one or more hydrogen atom. So therefore, the molecule of the unsaturated fat will be bent. Okay, or they have kink. So those are the answers that you can write for your answer. So the question worth only 2 marks, so you can give any 2 description. Okay, like for example, butter is saturated. Okay, oil, saturated fat lah. Oil is unsaturated fat. Okay, simple. So simple answer there for you. Okay.